Okay, so it's now 4 o'clock, so I would like to wish you very welcome to today's webinar, where I will talk about proteomics and metabolomics data analysis. And I'm Ann Holmberg from Qlucore. And if you have any questions during this webinar, please use the chat. So I'll try to answer questions while we go along, and of course there will also be time for questions after the webinar, and I will send you the slides after the webinar. So let's get started. So if any of you are new to the software, quick description is that Qlucore Omics Explorer is a very fast and easy to use tool to analyze and explore data without being a statistical expert. That being said, we also have statistical experts using the software, but the majority of our users are bench scientists, medical doctors, and so on that want to analyze their own data in an easy way. And to remind you, today we are talking about proteomics and metabolomics data, but we can analyze a lot of other different data types with the software. RNA-seq data, for instance, single-cell RNA-seq data, microRNA, qPCR, and so on. And there is a webinar tomorrow, which some of you already have registered for, regarding these two data types. And we support a number of different file formats. For metabolomics and proteomics data, we normally get the data in Excel and then convert it to TXT and CSV before we import it. So the agenda today is to first analyze a proteomic data set, which the title of that is Comparative Proteomics of Dying and Surviving Cancer Cells. It's a mass spec data set from MaxQuant from the Pride archive. And this has been published by researchers at the Karolinska Institute in Stockholm in Sweden. And the other data set is the metabolomic data set is called Sex, Steroids and Metabolic Dysfunction in Women. And that's also a public data set and published by researchers at the University of Chicago. So let's get started, and I will just shut my video like that. So let's start with the proteomics data set. So here we are looking at dying and surviving cancer cells. So when people are treated with chemotherapeutics, it causes the detachment and death of adherent cancer cells. And normally then when you study this, you look only at the remaining attached cells and ignore the detached cells. So this study tested the hypothesis that the proteomes of detached cells also contained some valuable information. So here we have the experimental design with 90 samples, 45 attached and 45 detached cells from 3 different cell lines and treated with 3 different drugs plus a control drug. So let's move to the data set. So I will open the software here. And here we are. So I will open this data set now from File, Open. And I will find it here. Here we have, you see it's a TXT file, which I open with a wizard. And I go to the next step here. So now I see the data set here. And up here we have information about the samples. So 6 different sample annotations. Here we have information about the proteins. So let's have a look. So here are the names of the proteins, as you can see. These are the variables, and here we have the actual measured data. And we can see directly that there are a lot of zeros in here, and this data is not log transformed. So this is something that we will take care of once we have imported it. So the first question here is this a raw count matrix, indicating that this could be RNA-seq data? No, it's not. So then we just move on. What's the separator? It's tab, that's correct. Are the rows variables or samples? They are variables, so that's correct. And then select the top left cell of the data matrix. This is the first measured value. And then select the top right cell of the data matrix. So then we go all the way to the right and pick that value. Click Next. Click on Variable Annotation Header Cells. This is the only variable annotation. Next, and click on Sample Annotation Header Cells. So here we are. If I press Shift and go to the last one, I take everything, and then we just press Finish here. Okay, so here we are. Good. So let's have a look in the table view here. And again, as we saw before, there are a lot of zeros and the data is not normalized. So let's go to the Data tab and use the Prefilter here and say that I would like to remove variables, remove proteins that has so zeros, so a value less than 0.1 in, let's say, 70% of the samples. Okay, and this, there are no rules regarding this. This is something that you decide yourself. But if there are so many zeros for a variable, I'm not that interested in that variable. So, and that caused, I originally had 5,500 variables, and with this prefiltration, I now filtered away 2,000 of them. Okay. I also want, and you see that there are much fewer zeros now. And I also want to log transform, and I also do that in the Data tab. Right now, the log here is grayed out, and that's because I still have zeros. I cannot log zeros. So I need to threshold to something low, maybe lower than 1. So 0.1, so something very low, lower than the lowest value in your data set. And now I can log transform the data. So this is normally a very good step to do before you start analyzing your data. So let's go back to the PCA plot, and now it looks like this. Okay, so we did a little pre-processing of this data. And I'm now showing my samples, my 90 samples here in a PCA plot, Principal Component Analysis. And now I want to have a look at the sample annotations. So here are all the sample annotations that I imported. So let's have a look at source. That's the attached and detached cells. So I color by that. And we see that, yeah, there are big differences here. And I can immediately see that there are bigger differences within the detached group than within the attached groups. And that's because the samples are further apart from each other here compared to here where they are closer together. But we can still see that there are a lot of subgroups within each of these groups. So let's perform a t-test now to see if we can find variables, proteins that are good at separating these two groups. So by selecting a t-test for the source, which is this annotation here, and compare one of the groups with the other group, and just set the Q value cutoff here to something low, like 0.1, for instance, 0.01, meaning that 1% of what I found here could be a false discovery, and that's very good. I might want to add a fold change filter, say that I only want proteins where there's a twofold difference between the expression between the groups, like that. So that brings me down to the 540 best proteins. And we still see that the blue group is very close together, and there is a big difference within the yellow groups. So if I now open up a new synchronized plot, let's color that by something else, and cell, for instance, here. So we can see that there is a big difference between the cell lines. Okay, so that contributes to the different groups we see here. There's also a big, if we go to the drugs, there are 3 drugs plus a control, and they are also behaving very differently. So maybe we would like to, if we go back and look only at this plot, maybe we would like to take away the effect of these, the cells, for instance, the cell lines, and maybe we also want to take away the effect of the drug when we are now studying the difference between detached and attached cells. So that is something that you can do using eliminated factors, as we call it, and these are also called covariates. So this is very easy to do in the software. And now if we want to have a look at the result, let's change this to a heat map. And so I configure the heat map by pressing auto here. I show everything, and then I can say that I want to hierarchically cluster the heat map here like that. And I might want to color according to source. No, sorry, source, like that. So we see that it's quite well separated. So if we compare with the PCA plot, we can see that there are indeed some yellow samples that are very close to the blue samples, which can then explain why this is in here. I can actually also use the flip, I think, to move that group. So if I flip this cluster like that, I actually tidy the heat map. And since it's a random order of the clusters, I'm allowed to do this. Yeah, so we can see that we have a lot of proteins here that are upregulated for the attached and downregulated here, and the opposite here for the detached cells. We can also add more colors to the heat map. So if I would also like to see the cell and the drug here, I can add that as annotations here. Okay, and then maybe we would like to study only the detached cells. So then we take away the attached cells like this. And then we need to change what we are looking at here. So let's say that now I would like to have a look at, so let's just go back like this. So I now have, so then I need to drag the statistical filter back to 0 in order to see things again. So if I color on cell line here, or let's do it the other way around. Let's have a look at drug, color on drug, and then do a multi-group test, an F test on drug. That. We can see that there are big differences. And now I might want to take away the effect of the cell lines. Like that. And then we can see that one drug seems to stand out here, the first blue one. That's very different from everything else. Like that. And we can also again have a look at the heat map to see how this like this. So these are examples of what you can do here. And you can, of course, always save a list of your results. So in this case, I have my resulting proteins here, and I can add the p-value, the q-value, sort on the column that I want. So I made a copy of the list here, and then I can say proteins separating drugs in detached cells, something like that. And then I can export this list, of course, like that. So that was a quick example analysis of this proteomic dataset. So I think I had a question, so let's have a look at this. Okay, so somebody's having problem with the audio. I have one question here. Can I do enrichment pathway analysis for selected groups of significant proteins? The answer is yes, but you need, when you do enrichment pathway analysis, gene set enrichment analysis, to have an ID match between the ID in your data set and the ID with the pathways that you are comparing with. So either you have pathways where you have a protein ID that matches your data set, or the other way around, maybe you have corresponding genes that you can enter into your data set and then compare with available pathways. But you need to find a common denominator between the two lists that you're comparing, the gene set or protein set and your data set. And I also get a question here. Can you name some of the largest databases for proteomics? And are the data in TXT file always? On the top of my head, I can only name the PRIDE archive, which was the example we had. And no, you cannot trust that it's always TXT. Data is normally uploaded in a number of different file formats. So this specific data set has also been pre-formatted by one of our experts before it came into TXT. So it's a wide variety, unfortunately, on uploaded public data. But if you are lucky, you might well find it in TXT format or Excel. Excel is easy because then you can just save it as a TXT file. Okay, any more questions? No, then we continue. Just when I send out the slides, I will also include a lot of the steps that I show you. So there are more slides in the presentation that I send to you than what I'm showing now. So now we move over to the metabolomics dataset. Here we are concentrated also on obstructive sleep apnea, OSA, which is traditionally associated with males, but it's under-recognized in women. This study points out that women with polycystic ovary syndrome, PCOS, who exhibits a shift in sex hormones, are particularly susceptible to OSA compared to women without PCOS. So this is an aspect that we are going to have a look at. So in this experiment, we have a look at the metabolome of men, healthy women, and women without polycystic ovary syndrome, with and without OSA. And there are a number of different interesting variables here that the authors are looking at, and we will have a look at some of them. So let's load this data set then. So then we close down the current data set here, like that. And we open up the other ones. So file open. Here we have it. And this has already been pre-processed and saved as a GE data file, which is our internal data format, basically a TXT file that we can open directly like that. Okay, and here we are looking at this data set. And here it's clear that we need to do some pre-filtering or something to get the data set open up. So what's very obvious here is that we have an outlier here. So let's see which sample that is. Okay, and we have a small diagnostic tool here, like that, which takes away, so the xval here takes away this sample, and the PCA plot is or the PCA plot is calculated without that sample, but it still remains in the plot. And we can see that when the PCA plot is calculated without this sample, it opens up. So this is clearly an outlier, so we need to get rid of that. So let's clear that again, because this is just diagnostic. I should not continue with that active. So instead I go over here and find this sample. So 9915. Here we are. So I just take that away, and then you see that the data set opens up. Okay, then we have a look in the table view, and again we can see that this data is not log transformed, and we can also see that there are a lot of italic numbers here, and that means that these are missing values that have been reconstructed. Okay, so I don't want to have a lot of missing values in my data set. A few missing values is fine, but not too much. So let's have a look in a heat map to see how much missing values we have, because the missing values are shown as black. And you can see that there are a lot of variables here that have a lot of metabolites that have a lot of missing values. So let's do a pre-filtration here as well. So go to the Data tab, turn the pre-filtering on, and specify what I want to do. I want to remove variables with more than 70% missing values. So a majority of missing values, then we take that metabolite away. And then we see here, we started with 6,000 metabolites, and now I have 3,800. And we also want to log transform, so let's do that. I don't need to threshold here because I don't have any zeros, so I can just log transform this data set like that. Oops, like that. Okay, and then we go back to the PCA plot. And we can have a look at the table again, and now you see I have log transform data. So values with, say, plus minus 25. I got a question, what does a missing value mean? A missing value is an empty cell in your data set, a value that has not been measured. So you don't know anything about that value. And then we need a value in that cell, so we reconstruct it. And the normal way of doing that, the default value is that we take an average for that variable for the rest of the actual values, that we make an average of them and give that value to the missing cells. We can also do a KNN impute, which is a bit more advanced missing value reconstruction. Okay, here we are. So let's have a look at the sample annotation. So gender, let's color on that. And we see that we have male and female samples. And we also have a sample that does not belong to any of those groups. So we take that away for the moment because that doesn't really help us at all in this analysis. So let's now do a t-test on gender. And we must pick one of the active groups. So male versus females, for instance. And we can see that it's quite easy to find either with the Q value of 0.01, like that. We can find 180 variables, or we can have a Q value of 0.05. That's also fine. And we can also filter to a fold change if we like, but then I go really far down. So the fold change of 1.5, I find 35 variables. So yeah, so that's an option that I have. So here we are. We can see that there is a big difference between the metabolome of men and women. So we can save that. So make a copy of this list and say men versus women, like that. And let's open up a new synchronized plot and have a look at something else here. We have this interesting annotation. So men that don't have PCOS, of course, women with PCOS, and women without PCOS. So let's color like that. And let's now make an F-test or a multi-group comparison on this annotation and try to separate these three groups from each other. So with a Q value of around 0.01, we see a slight tendency that the women with PCOS are moving closer to the male group. So this could mean that some of their metabolites are more similar to the male expression. So let's pull up a heat map and see if we can see this. I want to change this one to a heat map like that. And again, let's show everything. Let's hierarchically cluster like that. And let's color on this annotation here like that. Okay, and now, so this is quite common that it looks like this, that the clusters are not perfectly clear, and we can see that already in the PCA plot because the groups are not separating that well. So to make it easier to interpret the heat map, we can, instead of using the hierarchical clustering, which is unbiased, we can put a bias here and decide that I want the heat map to be sorted in this way. And then we can identify here, down here for instance, that it looks like we have a number of metabolites down here that are becoming more red. You see here we have the women with PCOS. Here we have the male group. So here we have a couple of metabolites that are becoming more red, are becoming more similar to the male group than what they are over here. And that's interesting. So let's make a list of those. So then I can, I maximize the heat map, and then let's say that I think that I have this cluster down here. So let's make a list of these genes here. So I guess it's that cluster. So then I go here to the variables tab, I create a new list, and then I use the list tool here and I pick these genes. So I think it's this cluster here. Oops. So now I picked these metabolites here. So you see I picked these 55. Then I tiler plots again. And now if I make this list the input list here, like that, now I'm only showing these 55 metabolites. And you see that the tendency that I saw becomes much clearer. Just to make so the pink women with PCOS are here moving even closer to the male group. So that's an interesting thing that you can find out here. And now we want to continue. So then it's very important that I take away this tick. So I continue now to use all the full data set again. And now we are going to focus on the other very interesting aspect here. So let's take away the men from the equation for the moment, because we want to study the OSA in women. So can we find a strong signal here between positive and negative OSA? Let's do a t-test on OSA here, positive versus negative. And I'm concentrating on the Q value, and I see that it's not really possible to find anything very significant here. Okay, let's then focus on only the women with PCOS and see if we can find anything there. So now I'm looking for women that has PCOS and see if I can find significant signal here. So it's not fantastic in any way. So with a Q value of 0.25, I can find 10 metabolites here, and we have them here. And among the annotations here, we have the KEG ID, which can be very useful. Okay, so it was not filled in here. That was a pity. But if we go back here a bit, you see that there are a lot of KEG IDs here. It can be useful when you want to compare with public lists. Okay, so I filter down again to my best ones here. Go up a little bit more. Can also add the Q value here, so we see what we have, and we can. Now I understand. Yeah, I must mark the active list. That's why I didn't see the KEG IDs. So now I see the KEG IDs, and I also see the Q values here, like that. Okay, so not a fantastic result, but still something maybe to continue working with. And now if I would like to, and I have the results here in the heat map, so let's just again do auto and auto so that we show everything, and here we have them. And let's also color the heat map on OSA. So here we have it. And let's sort it on OSA like that. And now I open up a new synchronized plot because I want to have a closer look at what I've found. So I want to make a box plot here. So I go to the method tab, all plots, and pick a box plot here. And I want the OSA on the x-axis, and I would like to see. So I pick manual variable here, and then I just go and pick either a complete list like this, or I can pick individual ones from down here. Like that. And let's also go into the options here and take away the p-value like that. So here we now see all 10, and we see that most of them, the positive is the pink one, most of them are upregulated, and we have one downregulated one here. So either look at many at a time or step through the list here and look at each of them like this. And then we can tile everything again like this. And now if I want to make a quick summary report on what I did, I can just go here to the log tab, PDF, and print a report. Desktop, summary, metabolomics. I can enter a comment. And here we are. So here are all the settings that I have applied to my data set. Here are all the annotations, all the samples that I've taken away, and some other information. And if we step down here, you see that here is my heat map with the annotations. Here is my PCA plot with the annotations. And here is the box plot. And here we have references for all the methods that are used in the software. So this is a really nice report, just to summarize what you did and also to find references. Yeah, I think that was everything I wanted to show you on the metabolomics data, and I see I have some questions, so let's see here. So a question here, hi, if we run two-group comparison, can we extract variables, list of up and down regulated proteins with fold chains more than 2 and less than 0.5 and get the number of proteins in each group? Yes, you can. So here in the statistics dialog, now I made a two-sided test all the time, but I can also say that I'm only interested in proteins or metabolites here that are upregulated, and then you will be interested in more than 2 here, or downregulated. So you can either look at the test two-sided, or you can do look for up or downregulated variables. What is the y-axis value in the box plot? So these are the expression values, but that's a good question because I wanted to show you. So we normalize to mean 0 and variance 1 always when we bring data in, because that works best with the PCA plot and the heat map. But when you start showing your data in box plots, then you can set the normalization to none and see the actual values. So these are the log transformed values. But you see that the normalization none is not very good for the resolution of the PCA plot, and it doesn't really make the heat map very clear. So for the PCA plot and heat map, it's good to keep the mean 0 variance 1 normalization. But when you move over to start looking at your results in 2D plots, it can be wise to set the normalization to none. And a question here, should we call it normalized protein expression? Now, so this is, I'm not sure what the correct term is. So this is a mathematical normalization that we do. So I guess that would be correct. But then, of course, we also talk about the platform-specific normalization. So it's important to distinguish between those two normalizations. Okay, so let's see where we are. We're almost done. So some more options that you have. Either you have a big data matrix that you bring in, but sometimes you also have individual result files. We can bring them in even if they don't have the same proteins in them. We will create the data matrix. The requirement is that the files must have the same layout. So that is described in the how to import data document in the reference manual. And you can, of course, also import your own list of proteins and metabolites for comparison, as I did when I created the list from the heat map and then used that as input. So you can do that if you have your own proteins and metabolite lists, but it must have the same unique ID. And as I got a question on also, you can perform enriched pathway analysis also with your proteins and metabolites, but you must then find either pathways that have protein and metabolite IDs in them, or find the corresponding genes for your proteins and the metabolites and enter them into your data set, and then you can perform pathway analysis. So that's the trick. You must compare with the same unique ID. Yeah, so that was everything from me. Are there any more questions? So while we're waiting for that, I can just talk a little bit more about the upcoming webinars. Tomorrow we talk about RNA-seq and single-cell data analysis. On Thursday, how you can make new discoveries by exploring your data. And then there are more webinars next week, including a basic training, hands-on exercise on Tuesday next week. And you register in the same place as you registered for this webinar. And if you are not already using or testing the software, you can download it from our homepage, Qlucore.com. Okay, and you are very welcome to contact me, or if you're already in contact with my colleague Jan, you can also contact him if you have any questions. So I would like to thank you very much for your time today. I will send you the slides. If you have time to answer the little poll that will pop up when you exit this webinar, we are very grateful for feedback. Yes, thank you so much and have a nice day. Bye-bye.